Assalamualaikum warahmatullahi wabarakatuh. My name is Yasmin Emilia Binti Rosli, and today I would like to present my final year project, Composition and Polysaccharides of Brown, Red and Green Seaweed as Anti-Cancer Agent. Malaysia is a country that contributes in seaweed business import to other certain countries. Malaysian seaweed was first started growth in eastern Malaysia, Semporna district, Sabah since 1978. The production of seaweed increased to 32% from 2008 until 2012. Seaweed farmed in Sabah dominantly by Carpophycus alvarezi and Carpophycus striatum, also known as cottony. The increased demand for seaweed and its derivative affords Malaysia an opportunity to further develop its position as a major global producer, with the Department of Fisheries Malaysia (DOFM) projecting production to reach 150,000 tons of dried seaweed. Seaweed are nutritional foods that rich in vitamins, low calories, and rich with mineral and other dietary fiber. Compound names alginate, agar, and carrageenan, not only popular among food industrial, also used as fertilizer and for animal use. Total annual value of production is estimated at almost $6 billion, of which food products for human consumption represent $5 billion. Some important compounds contained in seaweed give benefits in pharmaceutical areas such as carotenoid, polysaccharides, fatty acids, glycoproteins, haloforms, and some other compounds. According to Malaysian National Cancer Registry (MNCR), among 2012 until 2016, there were 115,238,000 new cancer registered. The number of cases increased by 2.3% in females and decreased by 0.8% among males per 100,000. Populations comparing with previous years 2007 until 2011. These statistical numbers prove that this disease does not easily cured with the existing medication methods such as chemotherapy, radiotherapy, hormone therapy, hyperthermia, immunotherapy, phytodynamic therapy, stem cell transplant, and surgery. These therapies also does not give hope to some patients as some therapy fails due to their health and how their immune system reacts to this medication. Most of these therapies only can help to slow the growth of the cancer cells and shrink tumors that causing pain. As these therapies left such side effects, seaweed treatment needs to be focused in medication as it comes from natural remedies and can cause less side effects compared to other therapies. This study was meant to enlighten the benefits of seaweed based on their polysaccharides concentration, based on their type, uses in medication, especially to fight cancer disease. This study helped to improve the cancer medication, though there are several other existing therapies. This seaweed study hopefully can improve and help to decrease the numbers of cancer patients where the natural remedies can be used as medicines such pills or dosage types. This review study intends to differentiate a few types of seaweed concentration by, first, to differentiate the concentration of polysaccharides of different types of seaweeds. Second, to find the most effective polysaccharides in different seaweeds for cancer medicine production. And lastly, to fasten the production of cancer medicine by using the highest concentration of polysaccharides from different types of seaweed. Are the following seaweed species that were chosen to examine their compositions. Uva lactuca, known as sea lettuce, are commonly used in European cuisine that it consists high nutritional values in terms of protein, carbohydrates, fatty acids compared to some nuts, grains, and fruits. Uva also reported use in agricultural utilization such as increased vegetative in bean plants, enhanced phenolic content, and help to enhance the antioxidant enzymatic activity. Cauliflower are very popular among Southeast Asians that it was consumed as low-energy food that rich with vitamins and minerals. These cauliflower are well known in Asia-Pacific region and was consumed as food, especially in Japan, Philippines, Indonesia, Vietnam, and Malaysia. In Thailand, These sea grapes are commonly used as sauce and salad dressing, make it most demand in current Thai market, which is approximately 1 ton per month. Color paracemosa extracts were several times tested by researchers as it was known have potential to use as sunscreen. It has bioactivation phenol that is an active ingredient to capture UV radiation. According to research, 1% extraction can have SPF value of 2.32, which can be used as the range of effectiveness of sunscreen cream between 2 until 4. It is well known used in biomedical application as it synthesizes gold nanoparticles significant for application in catalyst magnetic, biosensing, plasmonic, and pharmaceutical fields. Ketophore area was reported useful for dietary inclusion and have potential to enhance growth, immunological, and hematology. The sea area supplementation affects and enhances beta immune activation, directly affects health rates. Heteromorpha intestinalis extract used to produce melicite green (Mg), used mainly in aquaculture to treat parasites, has carcinogenic effect and genotoxic. E. intestinalis extract made chromium that used to fix dyes. The dry biomass then can be used effectively to remove the Mg and chromium ions from wastewater and environmentally friendly. Heteromorpha compressor polysaccharides potentially inhibit the herpes simplex virus (HSV) disease known as nootropic virus spread sexually transmitted. The derivative name SU1F1 is a sulfatate, show inhibition of virus absorption and virucidal effect. E-compressor extract in nanoparticles also used in biomedical application for its efficiency as antimicrobial and anti-cancer activity. This green seaweed table shows the difference of carbohydrate, moisture, and ash compound in different types of chlorophyta. These different results may be varied due to different origin or region which affected by humidity or any other circumstances. The highest carbohydrate is cauliflower which is 67.40%, and the lowest carbohydrate is E. intestinalis which is 34.28%. These results differ due to the different humidity, as cauliflower was originally from Malaysia with average 26 degrees, while E. intestinalis originally from India with average 21 degrees. Seaweed composition can be affected by humidity and temperature of the origin place and experimental place humidity. According to this table, the highest moisture is cauliflower which is 92%, and the lowest moisture is E. intestinalis which is 3.42%. This result clearly shows the moisture content affected by the origin humidity, as it also proved by research. Ash content shows the different result by different types of seaweed. The highest ash is Ulva lactuca, which is 26.99%, while the lowest ash is Chaetomorpha area, which is 7.45%. This can be affected by different methods used by researchers because of the humidity of Ecuador and India almost same, which are 90 degrees and 21 degrees. These are the following red seaweed species that were chosen to examine their compositions. Acatophora spicifera is a red seaweed distributed in tropical and subtropical areas and was made as food supplement in coastal region of Tamil Nadu, India. It has many potential as anti-tumor and antioxidant activity, also contain flavonoids and terpenoids as bioactive components. Centroceras clavulatum was believed can be extracted to use as antimicrobial properties and antibiotics of essential. It has potential to use as multi-drug resistant microorganisms to restore standard antimicrobial drugs. Capapicusalverizi var aring-aring is a species that contains high crude protein content and soluble protein, which functional ingredients in food products and plant-derived medicines. Capapicusalverizi var green tambalang has a potential on high phenolic content and microchemical during fermentation may contribute in development of nutrient food ingredients. Glaciaria corticata can be used to remove mercury from aqueous solution by bioabsorption using the dried red algae. This red seaweed table shows the difference of carbohydrate, moisture and ash compound in different types of red seaweed rhodophyceae. These different results may be vary due to different origin or region which affected by humidity or any other circumstances. It also may affected by different methods which are gravimetric determination or phenol sulfuric acid. By the researcher, the highest carbohydrates is kappa P kusalvarzi var aring which is 66.66% and the lowest carbohydrates is Glacyrrhea corticata which is 27.57%. These results differ due to the different humidity as kappa Phi kusalvarzi var aring was originally from Malaysia with average 26 degree, while Glacyrrhea corticata originally from India with average 21 degree. Seaweed composition can be affected by humidity and temperature of the origin place and experimental place humidity. According to this table, the highest moisture is with kappa P kusalvarzi var aring is 79.78% and the lowest moisture is with Glacyrrhea corticata is 3.98%. This result clearly shows the moisture content affected by the origin humidity as it also proved by research. Ash content shows different results by different types of seaweed. The highest ash is Centrochirus clavatum which is 36.69%, while the lowest ash is Glacyrrhea corticata which is 6.95%. This can be affected by different methods used by researchers because the humidity of Ecuador and India almost same which are 19 degrees and 21 degrees. There is diet supplement in market with Padina pavonica extract that can help to improve bone metabolism which is osteoporosis because research proved that it rich with amino acids and essential minerals. Spatoglossum shorteri can be the anti-human immunodeficiency virus HIV that infects with CD4 receptor at specific white blood cells. The polysaccharides have potential to inhibit the activity of HIV-RT. Sargassum polysaccharide is a species that gaining popularity over years in cosmeceutical formulations. Their polysaccharides derive antioxidant, anti-inflammatory, anti-wrinkling, and skin whitening effect. Harmophile succiniformis can be used as biofuel as it contains more unsaturated fatty acid compared to saturated acid, which shows the stability of biodiesel production. Dictyotadictoma can help to decrease heavy metal, copper and zinc ions in water pollution by bioabsorption method, which it will passive binding with the ions to take it out from aqueous. This table shows the difference of carbohydrate, moisture and ash compound in different types of brown seaweed, Phaeophyceae. These different results may be varied due to a different origin or region which affected by humidity or any other circumstance. It also may be affected by different methods, gravimetric determination or phenol sulfuric acid, by the researcher. The highest carbohydrates is Padina vaconica which is 43.39% and the lowest carbohydrates is Disotadichotoma which is 33.35%. These results differ due to the different methods used by researchers because the humidity of Ecuador and India almost same which are 19 degrees and 21 degrees. P. pavonica was examined by gravimetric determination while D. dichotoma examined by phenol sulfuric acid. According to this table, the highest moisture is Sargassum polysaccharide which is 83.51% and the lowest moisture is Dicotoma which is 4.23%. This result clearly shows the moisture content affected by the origin humidity as it also proved by research. Ash content shows different results by different types of seaweed. The highest ash is Spatoglossum shorteri which is 34.58%, while the lowest ash is Dictyotadicotoma which is 9.47%. This can be affected by different methods used by researchers because the humidity of Ecuador and India are almost same, which are 19 degrees and 21 degrees. There are a few types of polysaccharides that act as anti-cancer, especially alginate, carrageenan, and fucoidan. This type of polysaccharide is abundant in brown and red seaweed. Alginate is a versatile and adaptable biomaterial with a wide range of uses in biomedicine, including wound healing, medication delivery, and tissue engineering. Because of its small macromolecular structure, poor solubility, and low absorption, alginate's direct therapeutic effects in biomedical applications have been severely limited. Alginate oligosaccharides (AOS) as a bioactive small molecule has the potential to alter associated signal pathways such as MIR29B expression and toll-like receptor signaling. The expression levels of MIR29B to most cells in cancer patients were dramatically lower after AOS treatment, according to their findings. It influences the toll-like receptor signaling pathway, which includes a number of downstream components such as nitrogen-activated protein kinase. Carrageenan is a sulfatate linear polysaccharide that is gaining popularity as a food, pharmaceutical, and cosmetic substitute. It's also important in the development of diets, pharmaceutics, and cosmetic because of their biocompatibility, biodegradability, and low toxicity. The chemical structure of carrageenans is mostly regulated by a phase biosynthesis process that is affected by ambient circumstances, algal physiology, age, genus, and development stage. Alpha-carrageenan also reported to inhibit the growth of tumor cells B16F10 through intratumoral injection. Angiogenesis is a crucial stage in the tumor development. Carrageenans have been classified as angiogenesis inhibitors since they exhibit better anti-angiogenic activity than the standard chemical suramine. Fucoidans are a complex group of sulfatate polysaccharides found in cell walls of brown seaweeds, and they are the primary constituents of the sticky material found in brown seaweed. Native fucoidan from brown seaweeds generally has a diverse and branching structure. Other sugar monomers, including as mannose, galactose, glucose, xylose, and glucuronic acid, as well as a variety of sulfation patterns, are found in addition to fucose. Fucoidan normally has two types of homofucose. One type encompasses repeated 1-3 1-fucopyranose, and the other type encompasses alternating and repeated 1-3 and 1-4 1-fucopyranose. Fucoidan's anti-cancer potential may be regulated and mediated through a variety of processes and cancer-related pathways, including cell cycle arrest, apoptosis, and immune system activation. Some explore potential use of fucoidan in its soluble form to treat cancer, but recently fucoidan and base system developed nanosystem, the use of nanoparticles, micelles, or liposome as important role in biological system. In conclusion, seaweed is abundance of nutrients that can be provided into various chains such as food production, pharmaceuticals, biotechnology, nanotechnology, agrotechnology, cosmetic products, and others. Different types of seaweed have different composition based on humidity of each country's origin, which also give different benefits in various ways. Brown seaweed most likely to have the most abundance of polysaccharides, which are fucoidan and carrageenan, that help with anti-cancer and other medication. Each seaweed rich with benefits that should be explored more by researchers. The methodology that used by the researcher needs to be fixed, whereas some of the results of the seaweed composition are slightly different. The result may also be affected by the humidity and temperature differences between the countries. This brown seaweed needs to be explored more by countries with great humidity for seaweed production. This seaweed can be replaced or used by the medical team for cancer patients to lessen for any side effect. That's all from me. Thank you.